My name's Marlene Behrmann. I'm at Carnegie Mellon University in Pittsburgh. This is my email address. You should feel free to get in touch with me. I'm extremely grateful to be here today. I am so inspired by listening to people's stories and have learned so much from listening to all of you. Today I'm going to be talking about visual processing following resection surgery. In fact, the data that I will show you today is mostly from large resections rather than from hemispherectomy per se. This is just a little illustration of the sort of resections you can see. Each different color is a different kid, and as you go through the brain slices, you can see the extent and the volume of the resection. This is what I'm going to conclude. So if you don't listen to anything else, this is the message you should walk away with. It's early days in this research. There is some less good news, and I will share that with you. But there is also some more good news, and I will share that with you as well. And we still have so much to learn about the age of the surgery, the extent of the resection, the comorbidity of other neurological disorders, things about pre-surgical cognitive status that are all going to influence the nature and severity of the visual disorders that we will be seeing in these individuals. So if you're a 57-year-old and you have a lesion to this area of the brain, it turns out that you're really quite poor at recognizing faces. You may in fact think that these two are the same individuals. Actually, the projection here is not very clear, so it may even look to you like they're two different individuals. It looks like the same individuals, where in fact they're different. But if you're a 7-year-old with a similar kind of lesion that then goes on to have a pretty large resection, the question is, what is the functional outcome? Are you also going to show these kinds of visual processing disorders? And that's really the goal of the work that I'm going to be doing. So I'm going to show you data from about 10 such individuals. All of these have got lesions, resections, that affect the ventral occipitotemporal cortex, so the temporal lobe primarily, or the temporal and occipital lobe. And then I'm going to show you as controls individuals who also have resections, but it doesn't affect the cortical visual system at all. And we also have a lot of age and sex matched typical individuals who serve as control groups as well. So the real question here is, what is the impact, behaviorally and neurally, of posterior cortical resection to the visual system? About a third of the brain is involved in visual perception, all of these different colored areas. This is the left hemisphere, and we're looking at the occipital lobe right at the back. This is the medial surface, like if you were looking in from sort of the region of the corpus callosum, and you can see all of these different areas. So there is early visual cortex, which are these posterior areas right here. And then we have these more anterior regions that we'll call higher order visual areas. And I'm going to divide my talk into thinking about what happens after early visual cortex resection and what happens after higher order visual cortex resection. So these are going to be the two major themes that I will address. Let me tell you first about what happens after posterior cortical resection. So you undoubtedly all know that your kids have their vision screened using one of these perimetry tests. A light comes up. The kid says whether they detect the light or not. This child is hemianopic, does not detect any signals on the left-hand side following a right hemisphere occipital resection. And we can also look in these kids at what happens in their brains when they are shown images on the right or the left side of space. This is the left hemisphere, this is the right hemisphere, this is the occipital pole, and you can see that in a typical kid, you get a lot of responses in both hemispheres. So here is one of the cases who we've worked with. This is a kid with an left anterior temporal lobectomy. This kid has got full visual fields, as you can see here, no hemianopia, no field defect whatsoever. And when we look at their brain, this is the area that's been resected, this is the anterior part, but you can see their occipital lobe is responding beautifully. Suffice it to say that if you see any colors at the back part of the brain, they are responding normally and well. So we have typical bilateral maps in these early visual areas. And I'm going to show you a lot of data from these kinds of functional MRI studies. Here is a contrasting case. This is a child who had a right occipital and temporal lobectomy. This child has hemianopia, full hemianopia, robust hemianopia, on the side visual field opposite the resection, so in the left visual field following the right hemisphere. This is the resection. You can see a big chunk of cortex has been removed, and you can see that only in the structurally preserved hemisphere do we see any activation in response to visual stimulation. Moreover, this preserved hemisphere does not take on the role of the other hemisphere. It only is activated by stimulation in the right visual field. The left hemisphere does not respond to stimulation in the left visual field. You don't suddenly have a single hemisphere taking over the functions or representing information from both hemispheres. So the key issue is whether or not there is recovery from a visual field defect. And what is the chances? What is the chance of there being this kind of recovery? So we've done a series of studies. You don't have to read this. I just wanted to show you that we've got, line after line that details everything about these individual kids. They have hemianopia on the right or the left, sometimes just in a quadrant, and occasionally we see kids with full visual fields because their lesions are much more anterior. And in fact, we in our cohorts do not see any recovery of the blind visual field. We also looked in the literature because, of course, we don't have enough cases to be absolutely definitive. And so we wrote a very extensive review of the functional outcomes following lesions in visual cortex. And this is a review of pretty much all the reported cases that we could get our hands on. Only 4 of the patients in the literature are reported to have a recovered visual field. One of them is this case, AH, who was only born with a left hemisphere. And this single left hemisphere had the task of both the left and the right hemisphere of a control all kind of squashed into a single hemisphere. A very unusual, very atypical case with just a single hemisphere, but it was doing the job of 2 hemispheres. There are a couple of other cases from this Worth paper. The cases are unusual. Some of them have had surgery, some have not had surgery. They're at different ages. Some are prenatal, some are at day 2. And some of these kids are reported to have an improvement in recovery from the blind field. But I think the summary that we would probably want to walk away with is that there is no obvious, in the majority of the cases, reorganization of early visual cortex. We don't see activation from the blind field in the preserved hemisphere. Rather, we see persistent and stable hemianopia with very few exceptions in the literature. So that's maybe the less good news. So now let me try and share some more good news with you. So this is higher order visual cortex. So let me start off by telling you that in higher order visual cortex, I don't know how good this projection is, it looks a bit degraded from where I am, but this is early visual cortex at the back here, it's intact, and now we're moving somewhat forward to the, what I will refer to as higher order visual cortex. So there are regions in higher order visual cortex, say those shown in red here, that respond more to faces as stimuli than to any other kind of input. And there is a brown area over here that responds more to words than to other kinds of input. So we'll refer to these as category selective regions, responding to a particular visual category more than to other visual categories. And so the question that we have now is, if the resection involves category selective cortex, what is the consequence of this on the behavior of the child? So we do a lot of behavioral testing to understand what the nature of the perceptual performance is of these kids. Some of it involves our assessment of whether they can perceive global forms. So, for example, they have to tell us whether or not there is a stimulus that points more leftwards or points more rightwards out of these kinds of displays. Another way in which we do this is we show kids displays like this and we ask them which of these two has more swirl. The way in which you know that there's more swirl is you kind of trace a pattern through all of the local individual dots and you determine whether or not there's more swirl. You can determine that there is a form present from these little local elements. We also do other testing. Can they recognize faces? Can they recognize different kinds of objects? And here are the findings from a recent study. I think there were about 8 individuals. So these are the form perception. And all you need to see is that there are only 2 kids who, in red, so red means that there is an impairment, there are only 2 of the kids, somehow the asterisks have moved up, there are only 2 kids who show some kind of global form perceptual impairment, and it's the same 2 kids who are also impaired at face and object recognition. Think about this as more kind of pattern recognition. So for the most part, the kids are doing well, save for these 2, one of whom had other neurological comorbidities, in fact, polymicrogyria, and the other kid who was showing the abnormality is a kid with an IQ score of 40, so rather on the lower end. Okay, we can then look at what's happening in their brain. So this is what a standard brain looks like. When you put somebody in an MRI machine and you show them pictures of faces, houses, objects, some kind of scrambled, and then words. And we see this very prototypical pattern. This is early visual cortex in the occipital pole, as I mentioned. Then you can see large parts of the visual system that are activated by pictures of different kinds of objects. There are parts of the brain that are activated by faces, as you can see in red. Parts of the brain, a single part of the brain really that's activated by words, this so-called visual word form area in blue. And the parahippocampal place area, which is activated mostly in response to scenes or landscapes or buildings. So this is a kind of cartography of these category selective regions. So there are 2 green regions in the 2 hemispheres. This is the right hemisphere, this is the left hemisphere. This is how posterior versus anterior they are. You've got face selective regions in red, a visual word form area, object selective regions, and then early visual cortex. So this is going to be our shorthand for what the cartography looks like in kids after resection. So, wow, it seems to be really low resolution. I hope you can see it better than I can. This is the cheat sheet again. It's the cartography that I introduced, the typical signature. We have 4 of the kids who have absolutely typical and normal signatures, just like the typically developing individuals. These are the 4 kids where the resection is more anterior. So the category selective regions are spared in these kids. And in fact, we can define all 9 of these regions in all of these 4 kids. And then we have kids like this who have only category selective regions in the right hemisphere. And here is one who only has category selective regions in the left hemisphere. And we can then ask questions about whatever category selective regions we are able to identify. So let's imagine we can identify all 4 in some kids, all 9 in some kids, or just 4 in others. However many regions we can identify, we then go in and attempt to characterize the properties of the category selective regions that we've been able to define. We can look at how strong the response is in that area relative to control individuals. And I'll spare you all the statistical details. But suffice it to say, these 2 lines are the 2 horizontal lines here is the control data. And here are data from each of these individual kids. And you will notice that as long as we can identify a category selective region, those regions all fall within the normal parameters. So there is normal magnitude of selectivity. We can also look at what is the nature of the information that is being coded in those regions. What is its particular pattern? And how robust, how consistent and reliable is the activation of these regions over the first half of the testing versus the second half of the testing. So we look at the activation in the regions in the first half of the testing and in the second half of the testing, and we examine if a region responds really well to faces in the first half, does it also respond really well to faces in the second half of the testing? And how does this differ relative to typical individuals? And I will tell you that as long as we can identify category selective regions, they not only respond with the normal magnitude of activation, they also respond with the normal pattern of activation. They're essentially coding the same visual information as typical individuals do. So, in higher order visual cortex, if we define the region of interest with very few exceptions, we see a normal magnitude of selectivity. We see normal site of activation. It sort of obeys the topography of the brain. And it has normal neural information being coded in those regions. So you might ask, well, where does this kind of cartography come from in the first place? If you've only got a few regions, how is it possible that these regions come to be as normal as we're observing? And so I wanted to show you a little bit of data that we have from some of our longitudinal studies. Here are data from the kid with the DNet I showed you at the very beginning. We've scanned him very, very many times from age 7. He's about 12 now. Here is just a quick reminder. He's fully hemianopic in the left visual field following his right posterior resection. Here is the cartography, and I have to apologize that we used to use primary colors, primary yellow, reds, and blue, and somehow now my students have decided that they prefer pastel. And so it's the same thing, just in pastel colors. So this is the 4 scanning sessions that we have, and I'd like you to observe the following. This blue region and this green region is pretty stable from age 7 all the way through age 10. I hope you can see that it's sort of perfectly replicable in each of these sessions that we've been scanning longitudinally. The differences that we've seen is in these 2 areas, this more orange and this magenta area. The orange one is the word recognition area, and the pink one is the face recognition area. And we see that these areas in the single hemisphere that is preserved here kind of jostle for position over time and eventually come to have their own stable site and stable magnitude of activation. So these are the 2 regions. They're very close together in a single preserved hemisphere. And we can look at every single voxel, where a voxel is sort of some small area that we're able to scan. We look at all of the voxels in this entire region over the longitudinal scanning. And I hope you can get to see that over time, one area becomes more red and one area is beginning to be darker in blue. The more red it is, the more you see face selectivity. The more blue it is, the more you see word selectivity. So the single region in one preserved hemisphere comes to be specialized over longitudinal development to have both a word and face selective regions in that single hemisphere in adjacent positions, abutting each other. Okay, so we see this normal emergence of this topography in a single hemisphere over longitudinal development. The last small point that I'd like to address is some very recent work that we've done in trying to understand how the topography emerges. Is there a change in the connectivity between these different areas that make up this map that I've been talking about? We heard some earlier discussion about functional connectivity between different regions. I'm going to tell you about structural connectivity. We've also got functional connectivity data. And really the question here is there kind of rewiring? So we are going to look at, for example, these major tracts, white matter tracts, that go through the ventral occipitotemporal cortex. One of them is called the inferior longitudinal fasciculus. This more orange one is the inferior fronto-occipital fasciculus that goes all the way from posterior and swings round and ends up in frontal cortex. And we want to know what happens to these tracts in the kids, and do these tracts change over this longitudinal development. So we've only got a little bit of data. I think we've got about 10 kids so far. You can see in a control individual, we can see both of these tracts, one in green and one in purple. And this is a single resection kid. And here are some other examples with anterior resections and posterior resections. And wow, this projection is really terrible. It's impossible to see these slides. So I think I might have to just tell you verbally what the result is. So one of the possibilities is that if you have a resection, it will affect the entire tract. Not just the region of the resection, but everything else in that tract as well. You will get degeneration of the rest of the tracts. And what I can tell you is we do not see that. We only see degeneration in the region of the resection, and the rest of the tract remains intact. So that is the good news, is that we're not actually seeing sort of total dissolution of the neural circuitry in these cases. And we've got various other measures about exactly how the tracts work, and for the most part, if a tract exists, in the region where we can observe a tract, we can see pretty much normal tract characteristics. The circuit looks like it has integrity, it has the potential for signaling to other areas of the brain. And so again, there is some good news here that the circuits do not just fall apart and degenerate. Okay, so the tract is affected only proximate to the resection. There are some changes in the diffusivity in the way the signaling happens, but not very much at all. There is no obvious rewiring to the ipsilesional hemisphere, but the intact hemisphere is certainly carrying the burden of category selectivity and of visual perception and visual processing, and this is actually working pretty well for these kids. So let me just remind you of the take-home message. The less good news is that we are not seeing recovery of the visual field defect. When kids are hemianopic, they pretty much remain hemianopic, except for these very, very few reported cases. The some more good news is that the parts of the brain that are preserved in the cortical visual system appear to be working normally, have the same topography as we see in typical individuals, and is encoding the same kind of information that is true of typical individuals. The brain is not undergoing massive rewiring. It essentially is remaining stable. And it seems that the take-home message, most of all, is that the kids are making do pretty well with whatever brain regions they still have available to them that's kind of picking up visual perceptual abilities, and for the most part, their behavioral performance is good, and their neural profile is also, with few exceptions, in the normal range. There is much to be done here. This is undoubtedly all influenced by all of these variables that we've been talking about, all of these biographic, neurological, and demographic variables. I think the jury is ultimately still out on what the consequences of these resections are on visual cortex functioning. But I think there is a sort of glimmer of good news here, and that is that these kids are doing rather better than you would have predicted from just looking at the resection and the magnitude of the resection. I'll end off here by saying that we're running experiments on visual perception upstairs. I've got some of my students here with me. Many of your children are participating. We've almost got full slots for 2.5 days. We hope to take some really valuable behavioral data home with us and begin to understand what the distribution is of visual perception integrity versus deficits in a much larger group of individuals. And so I'm very grateful to all of you and very inspired by all of you. Thank you.